Our afternoon speaker this afternoon is Dr. Leland Lee Hartwell. He is the director, Pathfinder Center, Biodesign Institute, Arizona State University. And Dr. Hartwell led a research team at the genetics department, University of Washington from 1968 to 1997, studying the genetic control of cell division in yeast. He was president and director of the Fred Hutchinson Cancer Research Center from 1997 to 2010. He received the 2001 Nobel Prize in Physiology or Medicine. Congratulations. Currently, Dr. Hartwell does research in cognition, stories, and learning at Arizona State University with a team of undergraduate students. He has several honors. Some of those include the Albert Lasker Basic Medical Research Award, the Gairdner Foundation International Award, the Alfred P. Sloan Award in Cancer Research, the Genetics Society Medal of Honor, and the Caltech Distinguished Alumni Award. He is a member of the National Academy of Sciences. Dr. Hartwell, welcome, and we're super excited to hear your story this afternoon.
Well, thank you very much. It's really a great pleasure to be with you today. I am very interested and appreciate science teachers. When I first went to ASU, I spent some time actually doing demonstrations and activities in middle school classrooms and gained an appreciation for the challenges that you all face. As you heard, my current research is really focused on education. And I've been asking myself the question, how can we educate students for an unpredictable future? When you think about that, that's a tremendous challenge. It's hard to give students skills if you don't know what skills they're going to need. And I think we would all agree that we have no idea what skills will be needed 5 or even 10 years from now. So that's the big question for me. And in seeking some insight, I began by studying environmental problems and the need for sustainability. We created a course in sustainability education for teachers at ASU. And then I started studying sensory perception and cognition and the psychology of learning, and practiced what I was learning through my own courses at ASU. Over time, I've come to a set of very simple principles that I want to share with you today and hopefully get your thoughts as well. The first principle is autonomy. It seems to me that students must define their own values, their own interests, and their own goals. Every student has a different set of experiences, and I don't think there's any way we can or should impose our own values, interests, and goals on people. The second is the principle of agency. Students must take personal agency for their own lifelong learning. There's no way anybody else can do it. And so that's the second principle. And the third principle might be a little bit surprising, but I've come to it really through studying people in different careers, and that's the value of social skills. I find that social skills are the most important elements in career success, far more than the career-specific skills that we teach at the university. Well, how can we foster these principles as education? Autonomy, agency, and social skills. That's difficult. And that's where I want to think with you. One thing we can do is demonstrate respect for each student's process. That is, students may not be processing the material that we're teaching the way we expect them to, but they're processing it according to their own interests and values and goals. And I think we need to respect that, and that will encourage students. Secondly, we need to share our enthusiasm for the subject that we teach, because enthusiasm is infectious, and that can really stimulate students to develop their interests. And then the third principle is something that I really want to emphasize a little more later, and that's the use of stories or narration in teaching. Now, when I came to these principles after a great deal of effort, uh, I suddenly realized that it was like looking at myself in the mirror, because these are things I've always believed in my own autonomy and my own agency. And so that gave me cause to think about, where did I gain that perspective? How did I decide that my own interests were principal and that I was the agency of my education? And that question gave me thought to think to my own educational experience, and I came to a very new and deep appreciation for my past mentors and teachers that I had not really had before. So let me tell you a little about that. In terms of autonomy, I think of a physics professor I had in high school named Mr. Stoddard. Um, Prior to that course, which was in my junior year in high school, and I had switched high schools to a new high school, I had been a straight C student. I was an academic goof-off. I belonged to a car club, and I spent every night out carousing around and drinking way too much beer. Now, Stoddard did not lecture me or advise me, but somehow he discovered that I liked solving problems, and he would slip me difficult problems to solve. He didn't give them as assignments. He didn't give me grades or credit for them. He just thought I might find them interesting. And that was a tremendous impact on my life, because my grade average went from straight C's to straight A's. And I became engaged in academics as a result of a very subtle influence by this teacher somehow recognizing my interests and ability and feeding that for me. Now, in terms of sharing enthusiasm, I think of my uh, biology teacher at Caltech. I went to community college for a year after high school because my grades were mediocre, half A's and half C's. But I had a counselor at the community college, Glendale Community College, who cornered me one day and said, "We have a visitor coming through from Caltech, and I want you to meet with them." And I said, "Oh, I'm not interested in Caltech. I don't have any abilities for that, and can't afford it." And she said, "You're going to sit down and meet with this person." She just collared me. And it turned out I did get interested in Caltech, and I took the entrance exams and got in and became a physics major. But I took one course in biology just out of interest. And this was a course taught by James Bonner. And I can't remember a single thing I learned in that class. I don't remember us even using a textbook. I just remember Bonner down in the front of this lecture hall, day after day, jumping up and down, so excited about the future of biology because of the recent discovery of the structure of DNA. And he foresaw that that was going to completely revolutionize biology, and he was completely right. And his enthusiasm was infectious to me. I immediately changed my major to biology and never looked back. Then thinking about respect, when I was at Caltech, every semester I looked for an opportunity to do some kind of research. And actually, the research experience was much more interesting and influential to me than my courses. At one point, I was in the group led by Max Delbruck. Max Delbruck was a former physicist who decided to study life and began studying the very simplest forms of life, bacterial viruses. He later received a Nobel Prize for his initiation, really, of the field. Delbruck used to have a weekly meeting in which he liked to keep small. So he had it in a small room that would fit about maybe 20, 30 people at the most. And he would stand at the door and decide who could come in and who couldn't. And he would turn away full professors, but he would let me in. I was just a simple undergraduate who knew nothing, but that was respect. Delbruck acknowledged that I was a member of the scientific environment and that my presence should be respected. A postdoc in his group called Bob Edgar also showed me that same kind of respect and engaged me as a active member of the scientific discovery. One day I was walking by his office and he stepped out and he said, "Hey, Lee, come on in here. I want to show you something." And he pulled out these electron micrographs that he'd just gotten from Kellenberger in Sweden, I think it was. And they showed bacterial cells that had been infected by bacterial viruses. The viruses had mutations that prevented them from reproducing effectively. And what you saw in these electron micrographs were partially finished particles. And Bob Edgar realized that using this approach of mutants that couldn't finish assembly would allow him to work out the complete pathway of assembly of the bacterial virus. And over the next 20 years, he did that. And it was perhaps the most beautiful piece of work that has been done, really, in genetic dissection of complicated structures. But who was I that Edgar, with this new idea and this new data, would pull me out of the hallway, an undergraduate who didn't know anything, could offer no advice, and tell me about his discovery? Well, that was the kind of inclusion I experienced. Then let me talk about agency. After I finished my degree at Caltech, I went to MIT to work with Boris Magasanik, who was one of the few people in the US who was doing research on gene regulation. And I was interested in gene regulation, so I wanted to work with him. And I got there. I got a lab bench to work on, and he would come by daily to just talk with everybody in the lab. And I began to wonder when he was going to give me something to do. I expected that a graduate advisor would have a project for me to work on, but he didn't. And he never suggested anything. And I criticized him for this, later. Uh, and only later, only recently realized that that was part of his training. And so I had to find my own project, and I did. And he would keep coming by daily and just talk to me about what I was doing, what I was finding, never gave me direction, never criticized what I was doing, just showed interest. After about 2 years of this, and you can imagine the sort of um, pressure I felt coming by daily. I wanted a new result each day. That's a pretty fast pace. uh, Most advisors talk with their students maybe once a week would be frequent, once a month maybe more like it. So I tried to have a new result every day. After about 2 years, he came by once and he said, "Well, you've done enough for a PhD now." I had no inclination that I had completed that much work. And he said, "I can't let you go in 2 years because that's unprecedented, but you'll have to stay another year, but you're essentially done." Well, that was agency. uh, Boris Magasanik really expected me to become an independent scientist, to be able to do it on my own. And that was a tremendous training because, of course, a scientist has to be able to do it on their own, and they have to come up with a new idea every few years in order to keep the ball rolling. And then finally, in terms of encouraging my autonomy, I'm a very warm memory of Herschel Roman, who was the chairman of the genetics department at the University of Washington. Um, as a postdoctoral fellow, one usually goes to somewhere to learn the skills that you're going to need for your independent research. And after graduate school, I decided I wanted to study cell division because cell division was so important in biology and so poorly understood. And I think my background at Caltech had led me to understand that that's what scientists are meant to do, is pick something that's important and poorly understood and try to make a difference. When I took my first, when I did my postdoctoral research, I worked in Renato Del Becco's laboratory at the Salk Institute, and they studied cancer cells and they studied bacterial viruses that cause cancer in human cells and mouse cells. And I spent a year or year and a half there, and I became frustrated because I couldn't see any way to make progress in studying the fundamental aspects of cell division with the techniques that were available for human cells and mouse cells. So I left. I took a job at the new University of California, Irvine, sitting in my office trying to figure out what I was going to do. And I remembered back to Bob Edgar, and I remembered the uh, amazing power of genetics to uncover mechanisms in biology. And the same had been true in gene regulation. It was genetics that uncovered how genes were regulated in bacteria. And so I decided I needed to use genetics to study cell division. And I went to the library and started looking through the journals to see if I could find an organism simpler than human cells and mouse cells that one could do genetics with, but an organism that was higher than bacteria, that was more like human cells, that was eukaryotic, that had a nucleus and a spindle and chromosomes. And I came across yeast. And it turns out there is a rich uh, field of research in yeast genetics, but it hadn't reached the stage of cell biology in any way. It was just studying genetics and how genes were transmitted. I looked for the prominent people in the field, and one of them was Herschel Roman at the University of Washington. And so I called up Herschel and I said, I'm really interested in trying to use genetics to study cell division in yeast, and I don't know how to work with yeast. I wonder if I could visit and find out what you do. So he invited me up, and I spent just a couple of days, and he was so supportive. He sent me home with $1,000 micromanipulator that you needed to do yeast genetics. Just loaned it to me. He didn't know me from Adam. And that was the kind of support and agency and autonomy that I experienced from Herschel Roman. 3 years later, he called me up and offered me a job at the genetics department in Washington. And I came up and joined that department because I thought I could certainly use the influence of more geneticists around me and the help of graduate students who were interested in studying genetics. And so most of my career was at the University of Washington in that department with tremendous support from Herschel Roman for my whole career. So autonomy and agency, very important for science, for exploring the unknown. Now, that made a lot of sense for studying cell division, for studying yeast, for genetics, for the questions I was interested in, which is how does the cell achieve order in duplicating its chromosomes and segregating those chromosomes to daughter cells and then dividing. I was interested in how you establish the order of those events and how the cell assures the accuracy of those events and how the inaccuracy leads to cancer. That's the fundamental source of cancer is mistakes in that cell division process. And of course, it did make a lot of sense to be trained to take autonomy for my thinking and agency for my actions. I think now, with the tremendous uncertainty that even undergraduates face in their future career, that autonomy and agency are just as important for every career, not just for science, because we're all exploring an unknown future. So, coming back to that question of how can we encourage autonomy, agency, and social skills in our students? I think that in addition to respecting each student's process and in showing enthusiasm for their interests, that a third thing that we can do that is extremely important comes from stories. Through studying cognition, I've become aware of the fact of just how important stories are to our minds. Our minds are built for stories. We probably spend 50% of our time listening to and telling people stories. Almost all of our entertainment is about stories of other people. Through stories, we come to have vicarious experience. That is, our mind is built to what's called transport ourselves into somebody else's story, to imagine being the protagonist ourselves, to imagine having their experience. So that in the matter of a 15-minute, 30-minute story, we can get years of experience from someone else's personal experience. Stories are what make humans different than any other organism. It's the reason that we have technology. It's the reason that culture and science and everything advances. Stories are the way that we organize the past and the future and the present. It's the way that we relate events to one another by organizing them into stories. So, as I say, to me, our minds are built for stories. And I feel that education, certainly at the university level, has neglected stories, has neglected the power of stories. We've become so immersed in communicating information through textbooks that almost all of teaching is now expository and boring. We don't like to remember all these facts we're given in a lecture, but if we're given a story, we remember it. And now more than ever, with information so readily available on the internet, and even so well organized by AI agents, there's certainly no reason for us to be memorizing a lot of information. I think there's going to be, needs to be, a renaissance in using stories in education. My teaching now involves reading career stories with students, where we can experience what another person experienced in their careers. Careers are a set of challenges, one challenge after another, and success in careers means facing those challenges and developing a mindset or approach to solving problems in our career. And we can, through stories, participate in how other people solved their problems and developed in their career. Stories reveal the mindset that career professionals have developed over decades of experience. Not only that, but coming back to this point I made earlier, that social skills are more important than technical skills and something we neglect to teach. There was recently a book published called Gig a few years ago, which had stories from 150 different professionals, all different career stories. And in each of those, they sort of reviewed their career, talked about what they did daily in their life, what they liked about their profession and what they didn't like, and what problems they had. If you look at what problems they had, it's never technical. People can gain new technical skills as needed. The problems people face that they have trouble dealing with are social skills. And that's what we need to emphasize. And social skills are something we can learn vicariously from other people's stories. So these are the two reasons I think stories are really important. First, our minds are built to pay attention to stories, to vicariously participate in stories. Stories reveal how people solve problems and face challenges in their career. And stories reveal how to deal with social issues. In my own career, I was inspired. My entire research career was inspired by the story of Bob Edgar and using genetics to solve problems in bacteriophage morphogenesis. I used exactly that approach to study cell division in yeast. I used Delbruck's principle of taking the simplest organism that exhibits what you want to study, yeast, and using it. So it was, in fact, the stories that I learned as an undergraduate that led my entire career. Um, I think the best way to talk about stories is to give you an example. Um, there was a book published in the late early 1900s called Microbe Hunter. And this turned out to be a very influential book. And many of my contemporaries who went into microbiology and genetics and immunology and epidemiology were strongly influenced by this book. It's not a textbook, it's a book of stories. And let me tell you the story of the principles that were discovered for identifying an infectious disease organism. They were understood, that came to be developed by a medical doctor named Koch. Koch was a Prussian family doctor who used to travel around the small towns in Prussia to treat sick people. Of course, there wasn't a lot of treatment in that age, so it was primarily encouraging people to take care of their colds and uh, get sleep and take care of themselves. And he was pretty bored with being a country doctor. And so for his 28th birthday, Koch's wife bought him a microscope. Now, Koch had no scientific training. Science and medicine were completely separate in that age. And most people, I think, getting a microscope would look at pond water. You can see some really interesting organisms in pond water. Um, and that's not what Koch did. At the time, there was an epidemic of anthrax that was killing whole herds of sheep in Prussia, and even occasionally killing a sheep herder. So Koch trained his microscope on the blood of sheep. And he even had to create new microscopic techniques to be able to see well enough to see the little particles that were present in blood. And he invented oil drop microscopy. Now, this is a guy without any scientific training. And what he saw in the blood of sheep that were infected with anthrax was little tiny rods, very, very tiny bacteria. And their blood was full of these little rods. And when he looked at the blood of healthy sheep, they didn't have these rods. And so these became the first two principles of Koch's four principles, which is that to establish that an organism causes an infectious disease, you must show that the organism is present in all diseased animals, but not present in healthy animals. Now, that would have been enough. That was a huge insight, a tremendous understanding. And for someone who wasn't a scientist, I think most people would have stopped there, but not Koch. He had a tremendously rigorous mind. And what he decided was he needed to show that this organism was indeed responsible. And the third principle is that you must grow the organism in isolated culture and purify it. And so he took the fluid from an ox's eye and grew his organism in that fluid and found under a microscope, under his microscope, that the organism could grow in that fluid. And then he wanted to study the disease process, and he couldn't afford sheep. And so he needed to develop a model organism. And you'll recall the use of model organisms in biology in Delbruck's lab and yeast in my lab. But he foresaw this many, many years before us. And he decided to try mice because they were small and inexpensive organisms that he could keep in his laboratory, probably his kitchen. And so what he did was he he would take, there weren't needles and sterile techniques then, but he was aware of the fact that he needed to be sterile. And he would take a little splinter of wood and heat it to sterilize it and dip it in his ox blood with this little bacterial microorganisms and put it into the tail of a mouse. And the next morning, the mouse was dead. And when he looked in the blood of the mouse, it was full of these rods again. So he had shown that these little rods were alive because they could divide and reproduce, and he had shown that they could cause disease in a microorganism, in a laboratory organism, in mice. And that established the third and the fourth principles of cautious four principles, which is that you have to isolate the organism in pure culture, and you have to ensure that the pure culture will cause disease when re-inoculated back into the animal. And those four principles of COSH are still what guides epidemiological infectious disease research to this day. Now, COSH didn't stop there. The sheep herders told him that some fields were disease fields, and if they put healthy sheep into those disease fields, the fields would become sick and die. The sheep would become sick and die. But other fields were healthy, and they could grow their sheep in those fields, and they didn't become sick. Now, this seemed very strange to Koch because it meant that this infectious organism was overwintering the whole season, a year long, in these fields. And yet, the microorganism that he grew in his microscope slides in his laboratory were very, very sensitive. And if you dried them out, they died. And if you heated them, they died. And if you froze them, they died. And so, how could this happen? Serendipity appeared, as it often does in science, when he looked at an old microscope slide that he had put away. And what he saw was in these rods of his bacteria were little tiny round dots. And when he took these round dots and put them into the growth medium of the ox's eye, they reproduced and grew into rods. And so what he discovered was that the bacteria were capable of forming heat-resistant and drying-resistant spores, and that the spores were probably what was overwintering in the fields and keeping the fields infectious. And so he stopped the infectious disease spread by telling the farmers what you have to do is as soon as a sheep dies, either burn it or bury it before the bacteria can form these resistant spores. Absolutely brilliant work by a person with no scientific training. Well, I hope that story is interesting to you. I hope you think it might be interesting to your students. And I hope you might think about using stories to teach. Maybe you already do, and to excite the enthusiasm and interest of your students in a way that textbooks just cannot do. That's my lecture for today.
Awesome. Thank you so much. Utterly fascinating. And someone with a cell biology background, it's really cool to see a face with stuff that I've seen in a textbook, which is awesome. So one of the things that came up while you were presenting is, where did you end up finding this love for science? I know you ended up switching into biology, but like it seems like you kind of always had that. So like, especially those of us who are kind of K-12 educators, where did that kind of passion come from?
Well, as I got interested in science through solving mathematical problems from my physics teacher. So I intended to be an engineer and then a physicist when I ended up at Caltech. And as I say, I got really excited by the potential that our insights into DNA structure was going to have for biology. So it was James Bonner jumping up and down in front of the lecture hall that sold me on a life in biology science.
Awesome. We love those enthusiastic teachers, don't we? So in your years, especially since you have such an extensive history associated with both science and science education, what have you kind of seen from your perspective of how science education and instruction has changed?
Um, I think it has not changed at all. I think we are still using textbooks and we are still packing more and more information into every course because we feel like students need to learn all the facts. And I think that's anachronistic. The information is now available on the internet, and what we need to do is to teach students how to think, how to make sense of information, and they don't need to do that by learning everything. Those principles are relatively simple, apply to a vast array of different types of information. And so we really need to concentrate on how do we teach students to think.
Excellent. Completely agree. Speaking of stories, we would love to hear some of your stories as a researcher. What might be a story you have during your research that was something that was funny or kind of zany or weird?
Well, I think the most important story in my research was that I recognized the value of using a simple organism like yeast. I recognized the value of using a methodology of genetics to try to study a cell division that I was interested in, um, but I didn't know how to do it. And I isolated lots of mutants that were temperature sensitive, that could not grow at a high temperature, but could grow at a low temperature. So they were defective in some essential gene for reproduction at the high temperature. But I didn't know how to find the set that were important for cell division. Some of those were for protein synthesis, some of them for RNA synthesis, some of them for metabolism, for energy production. Where were the cell division mutants? And it took us 5 years to figure that out. And the way we figured it out, I guess it was more like 3 years to figure that out, but a terribly long time. And the way we figured it out was I had an undergraduate in the laboratory who was studying something strange. It's always fun to give lab undergraduates strange things to study. So we knew we had some mutants of yeast that formed strange shapes. And it had been shown in paramecium that if you did surgery to the cell surface, that the cells reproduced that surgical defect. That is, a surface defect was inherited. And I thought that was interesting. So we had mutants that formed odd shapes at high temperature, and the undergraduate wanted to see if those shapes were inherited. Well, we found out immediately they weren't inherited. And so we then wanted to know, well, what happens? How does the cell go from an odd shape to a normal shape? And so we said, well, let's look under the microscope. And as soon as we looked under the microscope, we realized that yeast grow differently than most cells. Most cells, when they divide, they just get bigger and then they divide in two. Yeast doesn't do that. Yeast forms a little bud on its surface, and the bud grows in size, and that's when the cell divides. So with most cells, you can't tell where they are in the cell cycle because they're just slightly bigger. But with yeast, you can tell because the bud gets bigger and bigger and bigger. And so that was the answer to how to find cell division mutants. We just had to look at the bud. I should have realized that 3 years ago. Why did it take me 3 years? Why did it take some serendipity of an undergraduate student studying odd-shaped yeast? Well, because science isn't smart. Scientists aren't smart. Scientists are just persistent. and depend upon serendipity to find their way. And it's really important to teach students that because they get intimidated when we teach them from a textbook that things are figured out in a very intelligent way, when in fact they aren't. The backstory is one of serendipity and goofing around and finding things slowly. Anyway, that's the story on how we got into cell division and finally found our way.
Oh, that's a wild story. Yeah, those things you just kind of walk into and ends up changing everything. So when it comes to doing research, I know you do kind of more of like the supportive role and whatnot, but what is your when you're kind of engaged in that research process, what's your favorite part of the process? What do you like to do the best?
Well, what I like the most is coming in the morning and pulling the petri dishes out of the incubator and seeing what happened. Um, Yeast is a tremendously useful organism because it reproduces so fast. It's a division time of about 2 hours. So a yeast cell can produce a whole colony of millions of cells overnight. And you can do experiments on individual cells in a matter of hours. You can do experiments on whole populations of cells overnight. And I think the secret to really having fun in science is working with an organism where you can have a rapid turnaround time, get experiments done quickly.
Okay, okay. When it comes to what you've seen with science education and whatnot, what are some things that are hopeful for? So knowing that, you know, where people are going with science education, what are some things that you were hopeful for?
Well, what I have discovered is that finding true career stories that can influence students, encourage them, but provide them accurate, real information is rare. It's hard to find true stories. Almost all the stories that we see and hear now, mostly from entertainment, are fiction. And so, how do we provide the true stories to students? And that's what I am really interested in now is I find my sources. I find um, mostly from memoirs of people, and lots of people write career memoirs. So there's a rich literature there. But you can't ask a student to read a full 300-page memoir every time you want to teach a lesson, right? So the problem is reducing those longer career stories to individual challenge stories that exhibit a particular principle that a person has in facing some challenge in their career. And it's creating those kinds of materials that now excite me and that I want to try to work together with teachers to develop materials that'll be useful in their classroom.
Awesome. That's so cool. What is your favorite topic to go through with students? Maybe it's like a lab or activity, or what is something that you really just enjoy engaging with?
Um, well, I think what I enjoy most is my dog. I'm really interested in cognition, and I'm very taken by the what language adds to and distracts from the way we think. And my impression is that dogs are just as smart as we are. They have the same size brain, are just as smart as we are in a nonverbal sense. And that um, I find that I'm actually more at peace when I am in my nonverbal thinking and just present in the moment, sensing what's going on. And I think that's what animals are doing all the time, and that we're not properly recognizing the incredible intelligence that they actually have because we are distracted by our language. So that's why I enjoy watching animals.
Oh, that's fascinating. I like that perspective. That's really cool. So continuing with stories, what might be a story from your past experiences, research or whatnot that was difficult? Something that ended up being hard, maybe you had to give up, something where you had to struggle.
Um, well, I guess the one thing is certainly the transition from basic knowledge to medical application. Uh, as you mentioned, I became director of the Fred Hutchinson Cancer Research Center. And the reason I took that position was because medical science is a combination of really three disciplines. Basic science that I had come from, clinical science, which deals with actual patients, and epidemiology, which deals with populations of people. And it's the integration of those three fields that leads to insights that are useful for patients. And I wanted to learn about that, and I hoped too that some of our insights from cell division and yeast would be useful. And so if you want to talk about what's difficult, I think the um, amount of effort and time and uh, experimentation that it takes to actually make a difference in the lives of patients is certainly a struggle.
Yeah, that definitely makes sense. So I saw that in general you do a lot of support for undergraduate and graduate students in their research. What are some projects that you've helped supported students in the last few years that really wowed you or amazed you with the work that some of the students that you were working with were going with their work research?
Well, I think what's been the most instructive is um, I have students who are doing their own research on their own careers. And so they're reading stories and trying to understand what they can understand from stories. But they're going beyond that, in that, as I say, the uh, availability of useful career-specific stories is limited. They're going beyond that to do interviews. So they interview professionals in their field. And interviewing is a very informative way to learn because you can ask people questions. You can hear what's important to them, and then you can begin to ask, well, what does that mean for me? Here are questions I have based upon your experience. And so I think the the magic in personal career research is in seeing what you can learn through interviews.
Yeah, that definitely makes sense. I'm a career and technical education teacher, so I hear you 1,000%. One of the questions that came up in our chat was, how might we support autonomy with our students when some of us are in situations where we don't have a lot of flexibility? Sometimes we have to do things in a very specific, segmented, sequenced way. How might we, any ideas or perspectives on how we might be able to support autonomy when we might be in those more structured environments as educators?
Yeah, that's probably the question, the question of our era, because uh, our history is one of trying to uh, teach to the consensus. It's presenting the consensus to the average student. And I know there's tremendous effort to try to deal with students individually, but there's no way you can do that with 30 students. And I think that's where AI comes in. Artificial intelligence really does present for the first time the opportunity of individualizing instruction. Now, I know there's all sorts of concerns about AI, and we have a lot of work to do in front of us in order to make AI serve our purposes and our needs rather than to take it at face value. But I think it is a tremendously powerful new tool to individualize education.
So related to some of the topics that you were bringing up, how might you see with your experience and whatnot, utilizing something like AI to support autonomy and agency and social skills?
Well, I do it almost daily. If I have something that I am thinking about, for example, this question of the role of language in cognition and the role of nonverbal cognition. Um, uh, there's probably a lot of people who have thought about that, and it would, if I used my old approach, which I did as a student, which is to go to the library and pull books off the shelf, it would take me a long time to access that information, right? If I went to Google, it would still take me a long time, although that's a tremendous advance. Now I can go to AI and I can get responses to my questions in a matter of a minute. I mean, it is unbelievable. And it's very stimulating. I don't necessarily believe everything AI feeds me, but I can have a conversation with it. I can disagree with it. I can ask, but what about? And actually have an intelligent conversation. Now, I don't want to speak for all AI. I'm using Claude from Anthropic primarily.
Okay, that definitely makes some sense there. So when it comes to social skills, one of the questions that came up is what might be the top 2 to 3 social skills that you might recommend for us K-12 educators to kind of focus on in support of their continued growth?
Ah, you found my weak spot. I never had any training in social skills. I don't know a thing about it. I just know it's needed.
Fair, fair. I totally understand. So another question that came up. So it's always one of those things that's curious, especially for our science-minded people. You found yourself with a Nobel Prize. You want to tell us how that story went, how you felt, that reaction, finding out that you were a Nobel Prize winner?
Well, of course, it's a tremendous honor, and I must say it never occurred to me that it would happen until just a year or two before it did, and I started getting a lot of awards, and then I started realizing that some of these were actually predictive of the Nobel. And but looking back, what I've got to say is that it's tremendous honor, but it's doesn't mean much compared to the actual experience of doing the science and interacting with students and learning day to day. I mean, that's where the fun is. The one thing I think that the Nobel adds is job security.
Oh, that's funny. That's funny. So, other than obviously, that particular glorious victory, what might be another one, getting back to those stories of something that you experienced that was, a moment of success, victory, that kind of stuff. Obviously, we know Nobel Prize is up there, but maybe another story of where you experienced victory or success.
The other thing that comes to mind is when we're getting very interested in this question of how the cell orders things. And it's essential, for example, that DNA replication and duplication of the chromosomes occurs before you try to separate the chromosomes. And we had observed research in animal cells that showed that a human cell, if irradiated, would arrest division. It wouldn't try to divide its nucleus. So somehow the animal cell was prevented from dividing because it had broken chromosomes. And so we realized that we could ask the question, why? And there were many yeast mutants known that were defective in repairing damage to DNA. And so we just looked at them to see if any of them were sensitive to damage because they divided when they shouldn't. So some of them were defective because they couldn't repair the damage. But we reasoned there should be some that would be defective because they didn't have the good sense to stop and wait and do it. And sure enough, it took just a matter of a few days to get the result that we predicted. So that was one of the few times when reasoning worked over serendipity.
Wow, that's wild. So it's such a quick turnover with response. That's wild. Um, so if you could talk to your elementary school self, what kind of advice would you give to your, just about to exit eighth grade self? What advice might you give?
I wouldn't give advice. I would ask the student, what are you interested in?
How about your high school self? Would it still be the same kind of?
Be the same question. And your interests will change over time. Your goals will change over time. Your values will change over time. And those are things we see from career stories, how people evolve and change their interests and their goals and their values. But those things have to match where you're putting your effort and your time. And if they don't, you're just not going to be very successful.
Yeah, that definitely makes sense. So for those that are into, we get into the college environment, it's hard to become a career scientist. Do you have some advice that maybe we can pass on to our students for those students that they definitely know they've got their interests, but becoming a career scientist is a challenge? Is there kind of some advice you might be able to give?
Um, I, I think it's probably true that many careers are difficult. They become much more scripted, and you have to do this, and then you have to do that, and you have to get publications, and you have to get grants, and so there becomes a a sort of checklist that you have to do. And this is true for many careers. It's true for medicine. It's true for all kinds of careers. And I think you you just have to ignore all of that and do what you really care about and find your way. And it may turn out that you really love science, but a research career it turns out not to be where your interests really are once you get into it, and you find that you like teaching, you know, or you find that you like writing, or you like being in museums or libraries. I think there are many different ways to uh, experience the things that interest you, and they don't necessarily have to be dictated by very uh, strict uh, career scripts that are, I think, too dominating.
Yeah, that definitely makes sense. So, continuing into the realm of cells, what are some kind of mysteries or discoveries that you think we're close to getting to, or where we might end up going, or questions that haven't really been addressed yet, in the world of cells and what you know?
Well, I guess I mean there's two things. Obviously, the ability to genetically engineer cells offers enormous possibilities, unlimited possibilities. We can, you know, I mean, you can imagine people with chlorophyll who can get their energy from the sun, okay? Anything is possible with genetic engineering. But I think the thing that interests me most from a, an applied perspective in the near future for medicine is advances in immunology. We're learning how to use the immune system to cure cancer, and I think it will be broadly applicable to many, many diseases once we learn how to really deal with what's a very, very complicated system.
Yeah, definitely. That is definitely true. When it comes to, and of course, many of us are going to be the fans of stories in science and science education. How might we as educators influence our various education systems, our districts, our schools, and whatnot to help us kind of advocate for that value of a story in the classroom?
I'm not one to give you advice there. I'm one to ask you how that can be done and how I can help. I'm see this as an important need for the future, and I'd love to be of help in any way I can.
Oh, then that leads into another one of our questions here. How do you see us as educators collaborating? What would it look like ideally? Is there, kind of a website or a system that you want to have set up so that way we can kind of interact and support?
Well, yeah, I think, as I say, the one thing that's really limiting is the availability of career-specific stories that will fit into the curriculum that you're trying to teach. So, you don't have time when you're teaching, photosynthesis, to bring up again, to go read all the background knowledge about how we discovered photosynthesis and what the stories are. We need to create a, a resource of great stories for the curriculum that teachers need to teach. We need to share those stories with one another. And maybe that involves not just written text, but maybe that involves audio and video and people telling stories who are good storytellers, and like TED Talks or like the Moth stories.
Awesome. That definitely makes sense. So heading back to scientific research, what is a task that you don't exactly like doing? Maybe you got to do it. It's not exactly a thing that you enjoy doing, but the job's got to get done.
Well, when I was a research scientist, getting grants, right? It's a very strange process because you submit a grant to a panel of peers, and they rank grants and fund the best ones. And that means that it's got to be something that is almost certain to work. And yet that's not the way science is done, right? And inventive science is done by doing things that you don't know are going to work. And so you have this constant process of submitting a research grant on something that you just discovered that you're sure you can now get some answers for, but hoping that 5 years from now, when you come up for the next grant, you will have discovered something unexpected that you can do it again for. So um, it's a lot of the formality of many careers is counterproductive to what's actually needed.
Yeah, definitely. That definitely makes sense.
I'm sure
So,
there's some of that in the teaching profession
oh,
as
ain't
well.
that the truth? Speaking of your kind of educator journey, you've taught at a variety of different entities, colleges throughout and universities throughout the world. What are some similarities and differences you noticed in that kind of experience, especially with your kind of international experience?
Well, I haven't had a lot of international experience. I mean, I've traveled, I've given talks at various places, but I think I could learn a lot, and we could all learn a lot by comparing approaches from different countries. And there probably are places where people use narrative and stories much more than I'm aware of. So I would love to learn more about comparative education, and I certainly don't have much experience there.
Okay. So I've got one last question. And so you have now a whole audience of educators in front of you. What would you love us to do? Is there kind of a set of instructions or a wish that you would like to impart on the several dozen of us educators?
Well, I think I'd like to help organize an collaborative activity in fostering the use of more narrative in education. And any of you out there who would like to participate in that activity, let me know, and we'll see what we can learn.
Awesome. Thank you so much.
So, Dr. Hartwell, thank you so very much. That was fascinating. I just, lots of it, lots of incredible good stuff. One of the things that teaching people to think has always been a big piece of mine. So it's kind of fun to hear that. So thank you. Um, Also as a thank you, from ASTA, we would very much like, we're going to plant 10 trees in your name. So we're going to send you a certificate for the 10 trees through the Arbor Day Foundation. And they have a mission to inspire people to plant, nurture, and celebrate trees. They believe that trees are essential for a healthy planet, as we all know, and planting and caring for trees is something that they have a passion about and they do all the way through. So we do have the 10 trees for you, and we will send you a certificate for that. And really appreciate all your time this afternoon and imparting your wisdom to us.
Well, that's a wonderful gift. Thank you so much. And the image you're showing here is exactly an image of the Northwest that led me to my career here. I think it was trees that drew me to the Northwest. Thank you so much.
Oh, you're welcome. And the Pacific Northwest is beautiful, so it's a great place to be. So thank you. Thank you.